The overall goal of the modified chromatin immunoprecipitation and DNA supercoiling experiments is to study the association and subsequent topological modifications induced by architectural proteins on site-specific DNA damage induced by chemotherapeutic or carcinogenic agents in human cells.
This methodology will allow us to address key questions about DNA damage and repair, which should facilitate discovery of new pharmacological targets for cancer therapy.
Our technology allows us to direct site-specific DNA damage and facilitates evaluation of critical DNA repair processes relevant to human cancer.
To begin, 24 hours before transfection, plate 400,000 mammalian cells per 60 mm dish in DMEM, supplemented with 10% FBS without antibiotics. Incubate the cells at 37 degrees Celsius and 5% carbon dioxide overnight. In a 14 milliliter round bottom tube, combine 30 microliters of room temperature transfection reagent with 500 microliters of 1X growth medium that has been warmed to 37 degrees Celsius. In a second tube, combine 2 micrograms of TFO-directed ICL containing plasmids in 500 microliters of 1X growth medium. Incubate the tubes at room temperature for 10 minutes. Combine mix 1 with mix 2 and pipette up and down to thoroughly mix. Then incubate the solution at room temperature for 25 to 30 minutes. Next, use warm PBS to wash the cells twice. Then add the transfection mix in a dropwise